Again, as we mentioned, the hormones, their cellular mechanism of action depends on binding to target cell receptors, absolutely. And based on their chemistry, these receptors are either going to be cell surface right or cytoplasmic. And that's going to initiate some kind of biochemical response. Turn on an enzyme, turn off an enzyme. Catalyze gene expression, don't, or whatever. And every hormone, every chemical messenger, not even a hormone, has a characteristic duration within which it is active. And we call that, the way we measure that is the half-life. This is borrowed from radioactive terminology. The half-life is the amount of time it takes for the initial concentration of a substance to be reduced to half. Okay. And that's a measure of how long it will remain active in our bodies. For that. And again, this is going to vary based upon chemistry, based upon structure, and things like that as well. The half-life will vary. We mentioned what they do. The answer really depends. Regulate rates of enzyme enzyme catalyzed reactions. They can open and close ion channels. They can synthesize or trigger DNA synthesis and the gene expression, or they can turn it off. It just depends on the exact scenario. Okay. Now, what I want to focus on here is the fact that how hormones work depend on their chemistry. And our textbook takes an approach of looking at 3 different categories, but I really want to look at it more not 3 different categories based upon what they're derived from, but I want to look at it based upon their chemistry, and this makes a lot more sense. Peptide or protein-based hormones, hydrophilic or hydrophobic?
Hydrophilic.
Hydrophilic. So water soluble, right? Steroid hormones, hydrophilic or hydrophobic?
Hydrophilic.
Hydrophobic. So these are going to behave differently, right? What about amino acid derived or amine hormones? It depends. What's the amino acid they're derived from? If they're derived from a hydrophobic, well, you have to realize, maybe you didn't know this, but some amino acids are hydrophobic, some amino acids are hydrophilic, some amino acids are acidic, some amino acids are basic, some amino acids are neutral, some are charged. So different amino acids, based upon their R group, right, have different chemical characteristics. There is a big split in amino acids. Some of them are hydrophobic, and some of them are hydrophilic. There really are only a couple different amino acids that hormones are derived from, tyrosine and tryptophan. One of them is hydrophobic, one of them is hydrophilic. So what these behave like depends on whether they're derived from tryptophan or tyrosine. And that will either give them properties that are hydrophilic or properties that are hydrophobic. Does that kind of make sense? So they're going to behave, things like dopamine and epinephrine, stuff like that. They'll behave based upon their chemistry. And they'll either look just like these and act just like these, or they'll act just like this. So we really need to know a little bit more about their chemistry to see how they're going to react in the body. If they're going to trigger, really if they're going to trigger a second messenger response, or if they're going to trigger what we call a genomic effect, which is where they actually get inside the cell and interact with